Welcome, and thank you for coming. My name is Satiro de Oliveira. I'm attending at Pediatric Hematology Oncology, BMT specialist. So our division of Pediatric Hematology Oncology is co-sponsoring this symposium together with CTSI, Lights Camera Cure, the Children's Discovery Innovation Institute, and the UCLA Gene and Cell Therapy Program. And the idea is actually to bring together a stellar group of speakers that will then try to show how feasible is cellular therapies for cancer and infectious diseases. Just a little bit of housekeeping. So we are webcasting this symposium to the satellite sites of CTSI, and all the presentations except Dr. Lawrence Cooper's will be available at the CTSI website for later. So we are recording the presentations. And so let's start. First, my disclosure. So I'm a pediatrician and I'm BMT doc. So for those of you who think cellular therapies don't work and are still stubborn, I will change your minds in a gentle but firm way. So going to the cardinal question, so what? So following the American Society of Gene and Cell Therapy definition of cellular therapy is the administration of live whole cells or maturation of a specific cell population for a treatment of a disease in a patient. So basically, instead of a molecule or a small molecule drug, what we have is a live cell that you're going to infuse into a patient. And as you can imagine, this is not a novel concept. Even blood transfusions it could be defined as cellular therapy, and certainly bone marrow transplantation. So that's why the stem cell transplantation is actually something that attends to that definition. But the idea is then, as technologies came through, the idea that you could harness immune effector cells, so then to use for cancer therapy, to treat infectious disorders, and then with gene modification of those cells, you could have these cells as carriers of a certain product, or to modify a target environment or a tissue-specific site. And of course, then biotechnology using cellular gene-modified cells or as scaffolds for multi-tissue organoids. And then, of course, induce pluripotent stem cells with the idea that you can then take a mature cell and reverse to then make a pluripotent cell. So why to do this? The idea is cellular therapies is patient-oriented. In that sense, you can expect fewer side effects. Patients with comorbidities would be then amenable to these therapeutic approaches, and in particular, transplant patients, you need to actually have them immune suppressed, or they will be immune suppressed because of the nature of the transplant. And because you are host-centered, the idea is you can streamline a certain processing line that then you can apply to different cancers with a single modification or to a different infectious disorder. And this way then it is the way to streamline processing for the so-called personalized medicine. So how to do this? And this is the question I wake up every morning until the time I go to sleep. And basically, you only see a lot of challenges, and that's why a lot of people will say that this is too difficult to come through. So the first big challenge is to even try to prove how safe and efficacious it is, the approach, and especially because if you want to use human cells, then you would have to use human models. So that is why then is the hardship of trying to devise the so-called humanized animal models where you can then study those cells. And of course, because this is a biological product, it is really hard to set the standards for optimal product and to define how many cells you need and how much gene modification you need for those cells. And of course, because of all these loose ends, then regulatory hurdles will be there, because you can't just take a patient and try whatever you want. Clinically relevant production of those cells, it becomes an issue, and our speakers will brightly bring that up. And as you can imagine, it's probably way easier to make enough cells to infuse into a baby than into a fully grown obese man. So you can imagine how hard it will be, and how different will be the processing for obtaining those cellular products. And because of that, then the development of those products and the production costs of all of that will be astronomical. And we are bringing Dr. Cooper, and he has the site from the other side, from the biotech company, to really explain how this can be done and what are the hopes for the future. And because of that, then all these hurdles and all these costs, then even if you prove it is efficacious and it works and it is safe, then how to use this in the clinical setting? When is the best time to use those approaches for our patients, especially for those disorders you already have standard of care practices? And how to bill for this, because of all the costs, and not all the centers can do it. So when? If you think about blood cell transfusions, we've been doing this since the 40s. And in stem cell transplantation since the 50s. UCLA actually was one of the first centers to have a BMT unit since 1973. Virus-specific T cells has been proven to work in clinical trials since the 90s. And we are still waiting to try to define how to build this through insurance companies. And gene therapy for SCID, and Dr. Donald Cohn is here, he's one of the pioneers of that. And it has been done, clinical trials for gene therapy for severe combined immune deficiency since the 90s. So everything culminated on then finally science recognizing cancer immunotherapy as the breakthrough of the year in 2013. And once again, even though everyone believes it works, we're still having a hard time on translating this to our patients. And another point for the when question is then, like I said, how to integrate this with for the therapy of our patients. So who? So the program for cellular therapeutics here at UCLA works in conjunction with the UCLA Gene and Cell Therapy and the pediatric BMT to try to then answer those questions and offer safer, less toxic, and more effective therapies for kids, because we are pediatricians. But the idea is to bring those concepts through, so then we can treat all the patients. Pediatric BMT is Dr. Theodore Moore, so he's actually the person trying to lead the whole thing. He is our division head for pediatric hematology oncology. And Dr. Donald Cohn, as I said, he's the internationally renowned gene therapist and a specialist in gene transfer technologist. And right now, I'm happy to say that we have then clinical trials and clinical protocols for treatment of multiple hematological malignancies and solid tumors through our BMT program, but also gene therapy for severe combined immunodeficiency, for CGD, adrenoleukodystrophy, and sickle cell disease. And for sickle cell disease, not only transplant protocols that are running, but also gene therapy protocol that is led by Dr. Donald Cohn. So we are bringing all these experts to then discuss about these issues and really try to answer these questions. Dr. Leen and Dr. Michael Keller will discuss about the approaches for infectious diseases, pathogens treatment, and really bringing first all the concepts and how to make it possible, and then discussing the current protocols. They're quite interesting and very innovative. Dr. Liao and Dr. Ribas, both of our rock stars here at UCLA, will then discuss about cellular therapies for cancer. And Dr. Cooper then will bring the point to discuss over those two approaches and mainly how to bridge this to successful translation, as he since May this year was appointed as the CEO of Zyopharm Oncology to really make all these therapies to come through. Just a little bit housekeeping rules. So the idea is all the speakers will have 35 minutes for the talk and 10 minutes for questions. If you guys don't ask questions, I will. And the idea is, because we are webcasting, the speakers will repeat the questions so that way we can have them recorded. Please sign in to make sure we have access to all you guys. And if you have any questions, please feel free to email me, and I'll direct all the questions, even for later on, to our speakers. The idea is really to foster collaborations and make sure we can have this coming through here at UCLA. And please also fill the evaluation. That way we can make sure everything is attending to your needs. And the idea is this is the first symposium of a series that we are going to launch, and hopefully later on then discuss more about immunotherapy approaches and induction of tolerance in transplantation. So that is why your input is quite valuable. Thank you.
Okay, it's a great pleasure to be here. I was just mentioning to the folks this morning, I've been through LAX many times, but I've never actually left the airport before, so this is a first for me. I'd like to thank the organizers for the kind invitation. Today, as previously introduced, I'm going to talk about the work we've been doing on virus-specific T cells. Okay. So, as many in the audience will know, viral infections remain a significant cause of morbidity and mortality, and this is particularly in the context of the transplant population. And for example, many of the patients that we deal with specifically are allogeneic stem cell transplant recipients. And conventional therapy with antiviral drugs is expensive, can be associated with significant side effects, and is often ineffective, which has led many groups, including our own, to look for alternative options to prevent and treat these infections. And of course, we're fixated on using adoptively transferred T cells. So what does adoptive T cell transfer look like in the allogeneic stem cell transplant setting? Well, what we do is we draw blood from the stem cell donor, isolate T cells ex vivo, expand out an antigen-specific or a virus-specific T cell population in the lab, and then infuse the stem cell transplant recipient. So we make one product from the stem cell donor, which is meant for use in a very individualized manner for that particular stem cell transplant recipient, almost a companion to the stem cells. And we've been doing this type of therapy now for many years. So this was originally started in the 90s by Clea Rooney and Helen Heslop, where they generated EBV-specific T cells. And this was in the pre-rituximab era, so it was to prevent and treat EBV-PTLD post-transplant. So that was a monovirus or a single virus-specific T cell product. We then extended the spectrum of viruses that we could target to include adenovirus and CMV. And I'm going to just walk you through our process for generating these trivirus or adeno EBV and CMV specific T cell products. So conventionally, we generated these cells by, again, starting with the donor's peripheral blood. And our first step was to infect the B cells with a lab strain of Epstein-Barr virus to generate EBV-transformed lymphoblastoid cell lines, which took about 6 weeks to make. Once this was established, we went back to the donor's peripheral blood and transduced the monocytes now with a clinical-grade adenoviral vector expressing CMV PP65 transgene. So now we're using the adenoviral vector backbone to stimulate adenovirus-specific T cells, and the transgene stimulates CMV-specific T cells. Then for the second and subsequent stimulations, we went back to the EBV-LCL and transduced it with the same vector. So now we have a B cell that's transformed and expressing EBV antigens, as well as adeno and CMV antigens. And in the presence of IL-2, repetitively stimulated the cells to generate a product in which part of the T cells were specific for adeno, part for CMV, and part for EBV. And again, this expansion protocol took about 6 weeks to do. And when we infused these into patients in this first trial, which was published now in 2006, so quite a while ago, we showed that these cells were safe post-adoptive transfer. We saw considerable expansion of the cells in vivo, particularly in patients who had active infections at the time of receiving the cells. We saw that these cells, these trivirus-specific T cells, were effective against all three of the targeted viruses. And I think this is notable because actually this was the first trial to show that adenovirus could be targeted using adenovirus-specific T cells. And we saw that the cells were effective in clearing disease in patients who had active viremia or indeed had virus-associated disease established. So I guess the question is, if it works so well and it's so good, and actually you can generate a broad spectrum product and adaptively transfer it to patients and provide that broad spectrum protection, then why weren't more people able to implement this type of therapy in their own centers? And so that brought us to look back at our process for generating the cells. And this is the schematic overview that you've already seen. And what we really felt that there were two major problems with our manufacturing processes. The first was the fact that, as you probably have all appreciated, the generation of these cells was really not that simple. So there were a lot of moving parts, live virus and clinical grade viral vector required to stimulate the cells. And overall, it took about 3 months to make every product. And then the second major problem to us, and what would really hamper this therapy being more broadly implemented in many centers, was the fact that this was really an ultra-individualized therapy. We were making one product from the stem cell donor, which was being administered to one recipient. And so for the next years then, so after this work was published, we really set about addressing these major limitations. And I'm going to walk you through the work we did to try to chip away at these problems. So first of all, to address the manufacturing issues. When we looked at it, we broke it down into three major parts. So we had detected the fact that in our multivirus-specific T cells, we were starting to see some antigenic competition. The more viruses we incorporated into our product, the more we saw that particular specificities were being squeezed out of our products, which would sort of limit the number of viruses that we could target within a single line. The next major issue was the fact that there was some significant cost involved in making these T cell products. And then, of course, that there was a lot of complexity. So these were three areas that we decided to focus on. So first of all, to show you what I mean by this antigenic competition problem. So as I mentioned, we started with single virus and then transitioned to bi- and trivirus-specific T cell products. And what we noticed is, as we added in more viruses, we saw that depending on the immunogenicity of the virus, there tend to be a dominant outgrowth of particular subpopulations of cells at the expense of others. And I think you can really see that very easily here. As soon as we added a CMV component to our T cell lines, the majority of the cells that we expanded were specific for the CMV component, and that really had an impact on the precursor frequency of adeno and EBV-specific T cells that we were able to grow out. And of course, although we could generate these trivirus-specific T cell lines, we felt that we were really locked in at 3. We really couldn't expand beyond 3 and potentially provide protection against other clinically relevant viruses. So to try to come up with an approach to overcome this issue of antigenic competition, what we simply did is look at supplementing our cultures with various cytokines that had been reported to prevent activation-induced cell death in our cultures. So what we thought is, if we could prevent virus-specific T cells from dying in our cultures, then we would maintain this broad spectrum specificity. But of course, the goal is here to expand and maintain the virus-specific populations and not maintain any cells that could potentially mediate alloreactive potential in vivo. And if we could find the right cytokine combination, this would allow us then to generate these nice T cell products with an increase in the frequency and repertoire of virus-reactive cells. And we looked at many cytokines, and here is just some of the data. So we looked at supplementing our cultures with IL-15 or 4 and 7, or even low-dose IL-2, and we compared that with our conventional methodology where we weren't supplementing our cultures with any cytokines at the point of initiation. And as you can see, in each of our cytokine conditions, we did see an increase in the overall number of cells that we're expanding. But the key is to know whether these are specific and whether we're preferentially expanding out T cells. So the first simple thing we did was look at the phenotype of the cells that we're expanding. And actually what we noticed very rapidly was that the combination of IL-4 and 7 allowed us to expand populations of cells that were both CD8, so cytotoxic, as well as CD4. And what I want to point out here is that in our hands, the culture supplemented with either IL-15 or IL-2 actually seem to drive more expansion of the NK cell subset. And while that's not necessarily a bad thing to adoptively transfer, when you're trying to provide T cell protection to your patients, you really do want to try to preferentially expand out a T cell population. And of course, as you'll notice with the cells cultured with IL-4 and IL-7, you don't grow out any NK cells. But of course, it's not enough just to have a phenotypic profiling. You then need to look at the functional capacity of the cells. And we wanted to see whether the cells we were expanding were virus-specific, and ideally we wanted to have a virus-specific population in both the CD4 positive and CD8 positive T cell populations. And again here, what was notable was that, What was notable was that only in the condition supplemented with IL-4 and IL-7 did we expand out a cytokine-producing antigen-specific population within both the CD8 compartment and the CD4 compartment. Whereas the IL-15 or IL-2 supplemented cultures, we saw specificity selectively within the CD8 population. And so with that, then, we thought that simply by supplementing our cultures with IL-4 and IL-7, we could reduce the problem of antigenic competition in our cultures. But what about reducing the cost? And again, when we looked at our overview of manufacturing, we felt that most of the cost really came from the fact that we had to use live virus and clinical grade adenoviral vector to use as an antigen source to stimulate our T cells. And of course, both of these are expensive to make and to continue testing. And so to look at alternative sources of antigen with which we could stimulate our T cells, we actually spent a lot of time looking at antigenic material, and we have indeed used clinically DNA plasmids encoding viral antigens. But the work I'm going to focus on today is our most recent work, where we've looked at whether we can use overlapping peptide libraries spanning immunogenic antigens from our target viruses to very simply stimulate T cells. And so, for example, if we were to try to generate a trivirus-specific T cell line with specificity for adeno, EBV, and CMV, what we would do is generate overlapping peptide libraries spanning immunogenic antigens from each of these target viruses. And in our original studies for EBV, we generated overlapping peptide libraries spanning two latent antigens, EBNA1 and LMP2, as well as the lytic BZLF1. For CMV, we decided to target PP65, but additionally also IE1, which had been published to be important for providing protective immunity in patients post-transplant. And for adenovirus, we focused on the two major components of the capsid, so hexon and penton. And when we very simply took these overlapping peptide libraries and exposed them to peripheral blood from stem cell donors, we found that we could very, very easily expand out populations of cells reactive against each of our target viruses. And so very easily then it looked like we could replace our live virus and viral vector with these overlapping peptide libraries. And then finally to the issue of complexity. And the majority of the complexity in our process really comes down to the fact that until quite recently, we have generated all of our T cell products using conventional 24-well plates, so the regular plates that everybody uses in the lab. And so just to give you an idea or a snapshot of what that entails in the GMP environment, so we generally start off with one plate of cells, we stimulate them, we expand them for about 10 days, we then harvest them, count them, replate them, re-stimulate them, feed them every 3 days, and keep reseeding, feeding, splitting, and so on, until we have enough cells required for infusion to each patient. And as previously mentioned, depending on the size of the patient, that can really sometimes be quite a challenge. And it ends up with the GMP technologist handling a lot of plates. And of course, that has its own issues with respect to the increased risk of potentially introducing some sort of contaminant into the culture. And so very simply then, what we did is look for an alternative vessel that would support the expansion of our T cells. And that really is easier said than done, because T cells are quite finicky and they really don't grow well in all plasticware. But after quite some searching and a lot of validation work, we came across these G-Rex devices. And what a G-Rex device is it's essentially a little flask, But the clever part is that it has a gas-permeable membrane at the base. And what this allows is for oxygen and CO2 exchange to occur right down here at the bottom where the T cells are growing. And so because the gas exchange is occurring at the bottom of the flask, one can add a lot of media supplemented with whatever growth factors and nutrients are required to support the T cell expansion. And you can add this all up front so you don't have to continually manipulate the cultures and the T cells continue to grow happily. And it is a static device, so you just pop it in the incubator, it sits there, there's no rocking and stirring involved. And of course, as I mentioned, because you can do all this up front, it significantly reduces the frequency at which you need to feed or manipulate your cultures. And in our original head-to-head experiments where we compared T-cell expansion in the G-Rex device versus our conventional 24-well plates, here's the growth profile. As you can see, the T-cells grow extremely happily in the G-Rex device, more than halving our production time. So this was our final point then that we thought we'd fix. Well, we have our cytokines to prevent activation-induced cell death. We have our peptides to use as a source of stimulation, and we've got the G-Rex to expand efficiently our T cells. But now if we implement all these different changes, the real question is, will they work? And will they produce a T cell product that is as clinically effective as our conventionally generated T cells? And that's really the big question. And so to ask that question, we developed a new clinical trial, which was called ARMS, for administration of rapidly generated multivirus-specific T cells. And in this study, because we'd overcome so many of the problems, what we decided is not just to target adeno, EBV, and CMV, but also target BK virus and HHV6. And these are the immunogenic antigens from each of these viruses that we generated overlapping peptide libraries for. So to stimulate the cells, all we did is pool these different peptide libraries together, expose these peptides directly to donor peripheral blood mononuclear cells, pop them into the G-Rex in media supplemented with IL-4 and IL-7, and expand them for just 10 days. So now it's just one stimulation and a 10-day expansion. And at the end of that, we have a multivirus-specific T-cell product specific for these target viruses. And I'll just briefly go through the clinical trial results from the first 11 patients that we infused on this study. Here, it's a standard phase 1 dose escalation study where we started at 5 million cells per meter squared, and our top dose was 20 million cells per meter squared. We saw no dose-limiting toxicity in these patients. One patient simply developed grade 2 skin GVHD, but this improved with topical steroids. Of these first 11 patients, 3 were considered very high risk for viral reactivation, and so their clinicians elected to infuse them prophylactically. And in all 3 of these patients infused prophylactically, they remained infection-free for at least 3 months post T-cell infusion. And that left us with 8 patients who we treated for active infections. So to go through those 8 patients, we had 2 patients who had a single viral issue that we treated. So 1 patient with adeno and 1 patient with BK virus. We had 3 patients with 2 viral issues. We had 2 patients with 3 viral issues. And we even had 1 patient who reactivated everything except adenovirus. And I think probably this is not really going to be a surprise to anybody who sees these bone marrow transplant patients, where really you can have one viral problem after another after another. So do they work? I'll go through again quickly some of the examples of the clinical responses with some of these viruses. So here's a patient who was treated for adenovirus. These are the weeks prior to T-cell infusion. So here you see the viral load exponentially increasing in the patient's peripheral blood. At this point, the patient received cytophelavir. It had some effect at the beginning, but again, the viral load begins to rise despite the continued use of cytophelavir. So at this point, the physician elected to infuse, and as you can see, this is what happens to the viral load. And this drop in the viral load corresponds very nicely with an immediate rise in the frequency of circulating adenovirus-specific T cells in this patient's peripheral blood. Here's an example of a patient who was treated for EBV-PTLD. Again, this is the picture in the patient's peripheral blood prior to T-cell infusion. So you see the viral load is exponentially increasing, and this is the patient scan showing quite significant disease. Here, at this time point, we infuse the cells, and as you see, there's an immediate impact on the viral load, just drops away. And again, this infusion of T cells corresponds with an increase in the frequency of virus-specific T cells. And as you can see, this is the post-treatment scan showing completely clear. Here's a patient who had a transient reactivation of CMV. So this occurred some weeks after the T-cell infusion. But again, this is controlled very rapidly. So the virus reactivates, the T-cells respond to that reactivation, they control the reactivation without need for antiviral therapy. And so up to this point, actually, what I've shown you is these T-cell lines that we can now manufacture very simply in 10 days are effective against 3 viruses that we knew were sensitive to T-cell therapy. So all we were trying to convince ourselves is that we could make these cells and we could make them faster and better, and they would retain their activity profile. But what nobody had tested before was whether BK virus and HHV6 were also sensitive to administration of adoptively transferred T-cells. So now I'll show you an example of a patient who was treated for hemorrhagic cystitis. This patient had high viral load, particularly high viral load detected in the urine, as well as the peripheral blood. And I should point out, we send our viral loads for testing at ViroCore, and 10 to the 10 is the max of their viral load assay. So we actually really don't know what the true viral load in urine was of this patient. We infused our T cells, and very nicely we see a drop off in the viral load. Symptomatically, the patient had improvement within a week or 2 of receiving our T cells, but again, there was an immediate impact on the viral load, which dropped away very nicely. And again, this corresponded with an increase in the circulating frequency of BK-specific T cells. And then finally, here's an example of a patient with HHV6. Again, this patient was infused up here for a different virus, had a transient reactivation of HHV6 3 weeks post-infusion. But what's really nice to see is as soon as the virus reactivates, the T cells sense and respond to that reactivation and begin to amplify immediately, controlling the reactivation. And so in this study, we defined a complete response as a return of the viral load to the normal range with resolution of clinical signs and symptoms, and a partial response to be at least a 50% reduction in viral load. And so with that in mind, to remind you, we treated upfront 8 patients for 18 different viral infections and achieved a 94% response rate with 15 complete and 2 partial responses within 6 weeks of infusion. And what I do want to spend just a moment on is our only partial non-responder, who is this patient right here. So this patient received a T-cell product and cleared their EBV reactivation and HHV6 reactivation post-infusion, but didn't clear BK virus. And we wanted to investigate why this was. So we looked at the donor T-cell line that we'd made and indeed found that the line did not have a BK-specific product, indicating more than likely that this donor was BK seronegative. And this will become important later on because our T-cell manufacturing process works very well when we generate lines from donors who have had prior exposure to these target viruses. But one has to remember that only about 70% of the population has been exposed to CMV, and probably 60 to 70% of the population has seen BK virus. And so while we can provide very strong immunity from donors who have had this exposure, for donors who are seronegative to target viruses, our manufacturing will not provide that particular protection against that particular virus, and that was the case here. Okay, so now if we compare our new rapid manufacturing process with our conventional manufacturing process, we're now able to target at least 5 viruses, and actually in preclinical studies we've gone far beyond these 5 viruses, so we can incorporate additional specificities for additional viruses, including respiratory viruses, which was something we're looking at quite a lot at the moment. We require just 20 milliliters of blood to generate these T-cell lines from donors, and our manufacturing is a 10-day manufacturing with a 7-day clinical release for use. And then finally, I do want to point out that using this 10-day manufacturing process, and it's a very small scale, nevertheless, we can really generate a lot of cells. So we can make about 400 million cells very easily with one of those little G-Rex devices, just using 20 milliliters of blood. And in the standard patient, we rarely require more than 1 T-cell administration, and probably the maximum number of cells that we would administer is probably 40 million cells. In the adults, so you have maybe an adult of 2 meters squared, so you're talking about approximately 40 million cells. So we make far more cells than we ever require for administration to a specific patient, which nicely segues on to my next part. So I hope I've convinced you that we can quite effectively reduce the manufacturing time significantly by implementing these changes that I talked about, peptide cytokines, using the G-Rex as a vessel for T-cell expansion. It still leaves us the problem, though, that we're generating this very individualized therapy. And of course, the holy grail for everybody is having an off-the-shelf T-cell therapy, which is exactly what we asked would be possible. Whether we could generate banks of cells that we could administer in the partially HLA-matched setting as an off-the-shelf therapy to treat these viral infections. And so to ask that question, we initiated a clinical trial. This is actually our second clinical trial, the first of which I believe Mike is going to briefly talk about in his talk, but I'll tell you about our more recent clinical trial, where we're investigating the therapeutic benefit of third-party T cells. This is, again, a product. So this is using all those additional cells that we made from our donor-derived T cell lines. So we have EBV, CMV, adeno, HHV6, and BK virus specificity. And what we do here, so for example, if I was a donor, and this is my HLA type, I'm HLA green, HLA-B red, and HLA-DR blue. So I would be a donor, I would go through the donor evals, and I would be cleared. We would generate a T cell product from me. We would aliquot all the cells, cryopreserve them. And the question we wanted to ask is whether we could then thaw these cells and administer these cells to multiple patients who are partially HLA matched with me. So, for example, this patient on the top who is HLA matched with me and who maybe has an adenovirus infection, or the patient in red who is HLA-B matched with me and has a CMV infection. So that was the idea, and at the moment it's a single-center phase 2 study. We are treating all 5 of our target viruses. And this is a 2-step process, clinical trial. So first of all, what we do is we get requests from physicians. So they say, hey, we have a patient, they have an adenovirus infection, and this is their HLA type, and this is the HLA type of their stem cell donor. Do you have a line that you think would be suitable? Then we go back to our database, which includes all the information of all the lines that we've generated, as well as the HLA typing. So we have HLA typing and specificity profiling that we do on all of our T cell products. We go through that, and then we will or will not have a T cell product. And if we do, we then proceed to the treatment part, where we actually administer that cell to the patient. This is a fixed dose study. Patients receive 20 million cells per meter squared as an initial infusion, and if they have a partial response, may receive additional doses at 2 weekly intervals. To date, we have infused 22 patients, and actually this is a very active protocol, so it really is probably more like 30 patients, but I will update you on the first 22 patients infused. We have had 16 patients who have required just a single infusion and 6 patients who have required 2 or more infusions. And this table summarizes the patients that we have infused. So the vast majority, as you'll see, were treated for a single virus issue. So these are the top patients here. All of these were treated for just one single virus. As you can see, by far, CMV and our BK virus are our more popular viruses. But we did have 3 patients who we actually treated for 2 viral issues. And again, showing the complexity of the patient population that we're trying to treat with these T cells. The first and major question, I think, when using any third-party T cell product is whether these cells are safe. And this slide summarizes the safety profile. So if we look first at acute GVHD, within 45 days of infusion, we just, again, had 1 patient who developed grade 1 skin GVHD, but again, this resolved rapidly with the administration of topical steroids. We had 1 patient who had a chronic GVHD flare, again, of the skin, and this occurred at the time when this patient's immune suppression was being tapered. So for our patients to receive our cells, we request that they have their immune suppression tapered to 0.5 milligrams per kilogram of prednisone or equivalent. And so at the time that they were being tapered for their immune suppression, they had a small they had a small flare of GVHD. So more than likely due to the tapering of immune suppression rather than the T cells. And then we just had 1 patient who developed a transient fever, but again, this resolved within a couple of hours. So do these third-party T cells work? And again, here's an example of a patient treated for CMV reactivation. I should point out, in this study, we are only treating patients who have drug refractory infections. So they have to have failed conventional antiviral therapy for a minimum of 1 week to receive our cells, or are simply unable to tolerate whatever the standard antiviral is. So in this case, this patient had CMV reactivation that was really persistent for a number of months prior to T-cell infusion. We infused our third-party T cells, and the viral load dropped away almost immediately. And again, this correlated again nicely with an increase in the circulating CMV-specific T cells post-infusion. Here's a patient with a CMV reactivation that had, an EBV reactivation that had been bubbling for a while. Again, patient was administered the T cells, viral load dropped away. Here the viral load is in the line. And again, this correlated with an increase in EBV-specific T cells. Adenovirus, here again, the viral load dropped away very nicely. And here's a BK virus patient. Again, this patient had hemorrhagic cystitis, and actually the majority of our patients on this study have either had hemorrhagic cystitis or nephritis. And again, this patient had a very nice drop in the viral load here, detected in both blood and urine. So of our 22 patients, again, we had a very high response rate, in this case 88%. Complete partial response rate in this study. And this is an ongoing clinical trial where we will hope to treat up to 50 patients all told. So with that, I hope I have convinced you that it is extremely feasible now to generate these broad-spectrum virus-specific T-cell products. The manufacturing platform is very simple and very robust. We do not have we have not had any production failures on any of our studies using this new manufacturing platform. We've made well over 100 lines at this stage. I hope I've convinced you that T-cell therapy in the allogeneic stem cell transplant setting is very safe, and I've convinced you that we can use either donor-derived T-cells or third-party T-cells to provide antiviral protection and therapy to patients. And then, just because this was kind of supposed to be more of a concepts talk, I just really wanted to give you a timeline of how all this work was done, actually. Because I've condensed everything into this very short talk, but it actually took a lot of years, and I started getting old when I was making this slide. I started feeling old, rather. So this all started in our lab with Cleo and Helen, who did all the work on EBV-specific T cells. And that was started in St. Jude's, and then they moved to Houston and continued the work there. So I was actually recruited to Houston in 2002. And I was recruited because they were having lots of problems with adenovirus in their children's population. And they needed somebody to study the immune response to adenovirus to figure out how healthy people control that virus, with a view to then taking that work into a T-cell product. And so I was recruited in 2002 and published a couple of papers in 2004 demonstrating that the virus was immunogenic and we could identify immunogenic antigens and so on. And that was then translated to 2 clinical trials. So the 2006 Nature Medicine paper, and then the Blood paper where we targeted just adeno and EBV, and that was for donors and recipients who were CMV seronegative. But while we were doing those 2 clinical trials, it became very evident that antigenic competition was an emerging problem, and plus the manufacturing. And I was actually making a lot of these cells that were being used in these trials. I was making them in the GMP, and the manufacturing was really killing me. And so that really prompted a lot of work to find better ways to make the cells. So we identified the G-Rex, we looked at alternate antigen sources, we looked at DNA plasmids, and combining DNA plasmids and antigen source with the G-Rex. We did another clinical trial in 2013 with that in the donor-specific setting. We also did the multi-center third-party study using the product that had been made using this technology to show that we could actually ship cells to different centers, including here, and they could be effective when other people thought them and administered them to patients. And then we continued on to try to improve it further. We looked at activation-induced cell death. We validated the PEP mix as a source of antigen. We wanted to incorporate new viruses, so we also did immune profiling for HHV6, which led to our 5 virus-specific T-cell product, and we've published that in 2014. As I showed you in the unpublished work, we're now doing the third-party study, and we have a lot of work that we have ongoing looking at new viruses and trying to make this bigger and better and more accessible for patients. And so that's where we are right now, and we're very, very busy. And then last but not least, I just want to show you our GMP, because people sometimes want to see pictures of our GMP. So this takes up a floor of our building. This is our GMP facility right here. our vector production facility here. Just to focus on here, my products are manufactured in these rooms, these 3 rooms. I have a lot of different clinical trials ongoing. These are some pictures from inside. This is the cord of the GMP. This is where we store our cells. This is just the gowning area where we get ready to go in. This is a typical suite. So the rooms where we grow our cells, they look like this. Actually, we're very fortunate because we have floor-to-ceiling windows, so we're kind of spoiled. And then these are some of the cell processing rooms where they isolate stem cells. And with that, I would just like to thank all these people. There's a huge team of people involved in all clinical trials, from the regulatory people to the manufacturing people to the clinical folks that help and refer us patients to all the folks in the lab. And actually, these are all the people that currently are working in my lab or have worked in the past to develop this therapy and get it to where it is now. And with that, I can take questions if there are any.
Yeah, my mistake, I missed your introduction, but actually you redeemed me very well. but it's really my opinion that Baylor is nowadays is actually practically dictating all the guidelines for cellular therapy production, and is actually one of the secrets why they are so successful. So questions?
Yes,
Oh.
Dan. Hi.
Beautiful work.
Thank you.
Of course, the question is, what does it cost? So, compared to the cost of having a patient getting antivirals, for toxicity,
Yeah.
can you calculate sort of what it costs for delivery?
You know, actually, that is what we've been struggling to try to cost for, I would say, probably over 2 years now, because it really is very hard to try to cost specifically how much it costs to get our antivirals, the additional days in the hospital, all this additional care. I can tell you that John Barrett has done some of this costing, looking at the difference that receiving a CMV-specific T-cell product makes versus, conventional therapy, and there's a huge cost saving. What I can tell you is currently our manufacturing costs for making these products, it costs us $5,300 to make a T-cell line, and from one line we can make product to administer to up to 30 patients. That's what we're currently allowed on our third-party T-cell study. And so if you think about the fact that we can administer a multivirus product, provide this protection, especially in the donor-specific setting where you provide these donor-specific T cells and they live long-term. You really provide this long-term protection to these patients and you're targeting multiple viruses. I think there's absolutely no doubt that there's a huge cost saving. And the other thing I would like to say is that what really has amazed me is particularly the BK virus patients. So anybody who's treated a little kid who's got hemorrhagic cystitis, I mean, it's just the most miserable thing in the world. And so, it's just so terrible for them. And they receive these cells and symptomatically they improve almost immediately. And so you'll go and do an infusion one day and 2 days later, the little kid's, playing, and, it's just the quality of life benefits are huge with these cells. But it's very hard to put, a cost on how much you save with these cells. But I'm a believer that there's a huge benefit to the patients of them. So.
Dr. Crooks.
Could you describe a little bit what the phenotype of these cells is when you infuse them? I would imagine from the way you described the regimen, these are largely a vector differentiated cells. Do they persist in the memory population?
They.
How long do they persist?
Sure. The question is, what is the phenotype of these cells and how long, or do we have any idea how long they persist? Yes. Yeah, so what exactly is the memory profile of these cells and, whether there is long-term persistence. So we do extensive phenotypic profiling. In the 10-day manufacturing cultures, we actually have about 40% of the cells that express CD62L. So they're RO positive, CD62L positive. And you have a population, depending on the donor, so there's some donor-to-donor variability, but you probably even have 10 to 20% that are CCR7 positive. And I think clearly there's, so the cells do persist, and I can actually conclusively say that because of Cleo and Helen's early EBV study. So the first 26 patients on that study received gene-marked cells. So they incorporated a retroviral modification, so the neomycin resistance gene, simply to look at long-term persistence of these cells. And they've seen these cells persisting out past 9 years post-transfer. So these cells have a very long-lived capacity, and you certainly are transferring memory to these patients. Any other questions?
You mentioned a little bit about EB virus and PTLD.
Mhm.
I'm wondering, during your clinical trial, did you see any graft versus tumor effect other than the PTLD?
So can you be more specific? I mean, we see a huge increase in the frequency of EBV-specific T cells and tumor clearance. But
when you try those kind of virus-specific T cells
Mhm.
to other kind of
Oh, so the question is whether we can use EBV-specific T cells to treat other EBV-associated malignancies. Is that so? This is work again that was. There are some ongoing studies, and actually even some studies that Kath Bollard was involved in when she was at the center. So we're using EBV-specific T cells to treat EBV-positive Hodgkin's and non-Hodgkin's lymphoma. And Steve Gottschalk at our center is using EBV-specific T cells to treat EBV-positive nasopharyngeal carcinoma, and he's actually collaborating with a group in Singapore now to increase the market size and the potential for that type of therapy. And I think Cleo is even thinking about using EBV-specific T cells for gastric cancer, but that trial hasn't yet initiated. So yes, these EBV-specific T cells have clinical benefit outside the setting of just PTLD. But of course, in the tumor setting, it's a little bit more complicated. For example, in some of the Hodgkin's patients that were treated with EBV-specific T cells who had an initial response to the treatment, their tumors relapsed and came back as EBV negative. And so that's a more general problem with targeting tumors, so that they will do a lot to evade the immune system. And so we have to keep trying to be better with our tumor-targeted therapies. Everything is just more immunogenic in the post-bone marrow transplant setting, so the tumor is very immunogenic and the T cells are very effective in that setting.
For your cytokines, do you have them all bought commercially available GMP grade? You
So
didn't
the
have to
cy-
produce
the-
any of yours.
No, yes, so the question is, are the cytokines clinically available? And yes, they are commercially available, yes. Okay, thank you.
So just to be clear, you can buy the G-Rex flask, but you cannot buy her cells. So now I'm going to introduce Michael Keller. Michael Keller is assistant professor at the Division of Allergy Immunology at the Children's National Health System. He's been there since 2013, yes. And his research has already been awarded the Young Investigator Award at the American College of Allergy, Asthma, Immunology in 2014, and the Translational Science Research Award from the Jeffrey Modell Foundation also in 2014. So that is why he's here. So please.
All right, thank you, Satiro, and thank you very much to the division for inviting me today. So again, my name is Mike Keller. I'm part of Dr. Catherine Ballard's group at Children's National Medical Center. And Dr. Ballard was formerly of Baylor, which you have heard from Dr. Leen has very clearly been the mecca of cell therapy for the better part of the last 2 decades. But we're very fortunate to have Dr. Ballard now at our institution. So over the next 45 minutes, I'm going to try to take you through a lot of the clinical trials using adoptive T-cell therapy to treat different infectious diseases. So I have no financial interests, and as Satiro mentioned, I'm actually not a transplanter. I'm a pediatric allergy immunology trained doc. So, and my specialty has mainly been in primary immunodeficiency disorders. So you'll probably see that little bit of a clinical skew through this talk. So I'm going to again discuss indications for T-cell therapy. I'm going to try not to overlap too much with Dr. Leen's talk, and then discuss the prior clinical trials of T-cell therapies, and then some current and upcoming trials. So as Dr. Leen mentioned, in patients who undergo allogeneic hematopoietic stem cell transplant, viral infections are both common and potentially serious complications. So data from PBMC suggests that they account for nearly 40% of transplant-related mortality. And in particular, the leading causes of viral-associated mortality are herpes viruses, such as CMV and EBV, as well as respiratory pathogens, including adenovirus, RSV, and parainfluenza. And then treatment of some of these viruses is available, but have significant toxicities, costs, and also resistance is increasingly common, particularly in the case of CMV and EBV. And certain populations are at particularly high risk of viral infection in the setting of transplant, such as those who have virus seronegative donors or those undergoing cord blood transplant. And also certain indications also impart high risk of viral infection, such as patients with primary immune deficiency disorders. So as Dr. Lee mentioned, adoptive T-cell transplant mainly stemmed from the use of donor lymphocyte infusion to treat viral infections. So this was very effective, but risked potentially fatal GVHD. So instead, virus-specific T-cells can be produced by expanding T-cells to specific targets over several days to weeks. And during this time, T-cells seem to lose alloreactivity and then can be given to a recipient with minimal risk of graft versus host disease. So there are several potential approaches to immunotherapy, and I'm going to talk about both of these options. So there's selection technology, and then also ex vivo expansion, which is what the groups at Baylor as well as we specialize in. Additionally, genetic engineering could also be used for targeting specific pathogens, and this could be either by using chimeric antigen receptors, transgenic TCRs. Additionally, resistant factors against pathogens can be added to CTLs, and there are current preclinical work going on to use this method. So current protocols for generating target-specific CTLs relies on either selection or cultured methods. So cell culture methods represent the majority of prior work, and in that, peripheral blood mononuclear cells are stimulated with the target of interest, eliciting proliferation of target-specific CTLs in both vectors and memory cells. And protocols use either viral peptides or transduced antigen-presenting cells against in order to stimulate peripheral blood mononuclear cells and to produce CTLs. So the current rapid protocol that Dr. Leen had talked to you about uses viral peptides and cytokines and can produce clinically useful volumes of cells in as little as 10 to 12 days. Alternatively, new methods also allow us to generate virus-specific CTLs from virus-naive donors, including cord blood or virus-naive adult donors, using donor-derived antigen-presenting cells and including dendritic cells and lymphoblastoid cell lines. The only downside to these is that the culture time required is fairly lengthy, with the usual minimum of 28 days of culture, and in total, roughly 6 to 8 weeks total of culture time. Alternatively, cell selection has also been used to target specific viruses, and this has been used in the past either via multimer selection for specific viral epitopes, or using stimulation with a specific viral epitopes followed by column selection targeting either interferon gamma or CD40 ligand using FDA-approved devices such as the Clinimax device. The advantages there, obviously, is that it has a much shorter production time. It can be made in as little as 1 day. However, both the rapid protocol via culture as well as cell selection requires that a donor have prior immunity against the viruses of interest. So that is obviously one downside. So following ex vivo expansion, virus-specific CTLs have specificity against the targeted epitopes with both class 1 and class 2 restrictions as shown here. Here you can see that predominant CD8 skewing of the CMV response and a predominant CD4 response, on the other hand, against EBV as well as adenoviral hexon and penton. And this is fairly typical for many of the donors that we see. So the first question in clinical trials, obviously, that we have to address is whether or not adoptive T-cell therapy is safe. And the obvious concerns here would be whether or not there's any induction of graft versus host disease or cytokine release syndrome. And to date, the vast majority of studies have shown little to no attributable increase in graft versus host disease after CTL infusion. And this was summarized quite beautifully by Jan Mellenhorst and colleagues at Baylor in a prior blood paper. In most cases of GVHD that occurred after CTL therapy, as mentioned, mostly occur in the setting of weaning immunosuppression in order to allow these T cells to be efficacious. And rare cases of de novo GVHD that occur in the first 45 days are uniformly treatment responsive. And secondly, are CTLs effective in preventing or treating viral infections? So cytomegalovirus was certainly the first virus to be targeted by adoptive T-cell transplant nearly 20 years ago. Since then, over 200 patients have been treated with CMV-specific CTLs on various protocols, and the vast majority have achieved either partial or complete antiviral responses. Cell doses have varied pretty widely, but with the current protocols being anywhere from 5 million per meter squared to up to 1.5 times 10 to the 8th per meter squared. But seemingly the chances of an antiviral response do not seem to correlate really well with cell dose, and most trials have now landed on a 20 million per meter squared dose, which seems to be effective in most cases. Many failures that have occurred usually are associated with requirements for high-dose steroids in the setting of graft-versus-host disease. Additionally, it's possible that CMV factors may contribute to some treatment therapies, and currently we're studying whether or not certain escape mechanisms may potentially play in to the rare treatment failures. So as Dr. Leen also mentioned, if you give adoptive T-cell therapy derived from a stem cell donor, they tend to last a very long period. And this was shown basically in prior studies, in the fact that patients who received donor-derived CTLs had a much lower rate of late-onset CMV disease. And even in those who had viral reactivation, antiviral medications are often unnecessary, as usually there would be a transient reactivation and then a clearance of the virus concomitant with the rise in CMV-specific TCLs in the peripheral blood. Multiviral CTLs are also effective at treatment against adenovirus, and in Dr. Leen's 2006 paper, she and her colleagues described 5 patients who had active adenoviral disease at the time of infusion, including 2 who had fairly significant respiratory disease, and all were able to clear disease after adoptive immunotherapy. And here you see again from their paper the CMV viral count in black dropping off as they actually were able to detect adeno-specific T cells in the peripheral blood of the patients. So EBV also can be targeted by CTL therapy, and in the Baylor cohort, EBV-specific T cells provided near perfect protection against EBV-PTLD. And in 11 patients with existing PTLD at the time of infusion, CTL therapy resulted in resolution of disease, as shown in this imaging of hepatic PTLD, which you see in the imaging here, which resolved within 2 months of adoptive immunotherapy. Some of these early cell products also had genetic marking and were detectable up to 105 months out after infusion. So here's a summary of notable phase 1 and phase 2 protocols targeting CMV, EBV, and adenovirus. The top 3 here were all via column selection technologies, whereas the latter 6 are by ex vivo expansion. In essentially all studies, preventative therapy has been highly successful. Treatment of existing infections is also very effective in the majority of these prior studies. And the rare treatment failures, particularly against EBV, were usually in the setting of fairly bulky disease or in the setting of high-dose immunosuppression. So I'll talk a little bit also about our work at Children's National since Dr. Baller arrived. So we've treated to date 18 patients at Children's National plus 4 external EINDs with a variety of age ranges. As you see, our indications have skewed a little bit toward non-malignant conditions, which likely reflects our referral base and our skew toward referrals for primary immune deficiency. And to date, of those patients, 12 had active viral infections at the time of infusion. And we had, so far, 85% response rate against CMV, and perfect response rates against EBV and adenovirus. Again, as was seen in prior papers, efficacy tended to correlate with detection of these T cells in the peripheral blood of patients in vivo. So here is actually patient number 1 from Children's National, who was a baby with SCID who had chronic CMV and had undergone a non-conditioned haploidentical maternal bone marrow transplant, but still had ongoing CMV post-transplant. So, and the CMV was already confirmed to be resistant against ganciclovir and foscarnet, and the baby came to us actually on sodopavir still. So he got 2 doses of maternal-derived multiviral CTLs at 5 million per meter squared, and ended up needing that second dose because of immunosuppression. But you see in red, his CMV viral count dropped fairly precipitously thereafter. And at the same time, over the months after therapy, we were able to detect anti-CMV T cells in his peripheral blood. So he is now over 1 year out from transplant and doing quite well. And here's another patient. This one was a 1-year-old with Wiskott-Aldrich syndrome who underwent matron-related bone marrow transplant and had EBV reactivation in the first month after transplant. He had received rituximab, still had EBV viremia, and then received a single dose of donor-derived multiviral CTLs that targeted LMP2 and EBDA1. And you see that within the 7 days after receiving those CTLs, his EBV viral count dropped off precipitously. And again, as in previous cases, we were able to detect T cells targeting LMP2 and EBDA1 in his peripheral blood, which expanded and then contracted physiologically in the month thereafter. So one additional methodology that we're trying to use at Children's National to classify our responses to T cell therapy is by TCR beta sequencing. And that's been used very successfully to classify T cell repertoire and also reconstitution after bone marrow transplant. So we've been fortunate to have access to it to classify and track CTLs in our subjects. And the concept is essentially to use ultra-deep sequencing of the CDR3 region of the TCR beta receptor to identify specific T cell populations that can be tracked longitudinally. So here I'm showing a patient who had CMV reactivation, and as shown, he didn't have this particular TCR beta clone pre-infusion, and then after infusion at 1 month, he had a rapid increase in the specific CMV-specific clone, which was detectable greater than a year out, and it expanded concomitantly with drop in his CMV viral count. And by utilizing cell sorting, utilizing either MHC pentamers or functional assays, we can identify virus-specific T cells, as shown here, and then particularly then go on to sequence these populations in order to tie specific TCR beta sequences to viral specificities and MHC restrictions. And we've been able to do that for a variety of dominant CMV epitopes, which often have a small number of highly dominant CDR3 sequences. So this allows tracking over time in patients who receive these CTL therapies. So here we see CDR3 sequences corresponding to dominant HLA-A02 PB65 epitope called NLV. And these 3 sequences show long-term persistence for up to 3 years following infusion. And here I'm showing in these 3 different markers, 3 different patients who are all HLA-A02. So returning to recent clinical trials, there have also been 2 protocols that have recently shown that expanded viral coverage is feasible and can also allow targeting of HHV6 and BK virus, as described by Dr. Leen, as well as varicella. So here, Dr. Papadopoulos and colleagues at Baylor, as Dr. Lee mentioned, treated 11 patients with 5 virus CTL with overall excellent response rates, even when targeting multiple active viral infections with a single product. So they noted that, as mentioned, the one patient who failed to clear BK was likely due to a donor who was BK naive. And also Chun and colleagues in Sydney also reported treating 10 patients recently with 4 virus CTL, so that's specific for CMV, EBV, adenovirus, and varicella. And they actually used the Varivax vaccine to infect donor-derived dendritic cells in order to produce those varicella-specific T cells. They also saw good protection against CMV, but they commented that they couldn't make much of the VZV component given that there were no clear exposures to varicella and that all patients remained on acyclovir prophylaxis throughout the treatment course. So both at Baylor as well as at Children's National, we've also been working to expand the number of viruses that we can target via a monoculture. And we've recently here I'm showing data targeting 8 different viruses. So that's CMV, EBV, adenovirus, HHV6, BK virus, RSV, H3N2, and varicella using PEP mixes in a rapid protocol. And so it clearly actually is feasible using a rapid protocol. And overall, from five healthy donors, we were able to elicit specificity against a median of 6 viruses and 14 of 17 epitopes. So we are unfortunately limited to what the donor is immune to, but seemingly, especially for viruses that are very ubiquitous in the population, this is possible by a monoculture. So with respect to indication-specific responses, we recently reviewed the use of virus-specific CTLs for primary immunodeficiency disorders at 4 centers, Baylor College of Medicine, Children's National, and then Newcastle and Great Ormond Street Hospital in the UK. And in total, we looked at 36 patients with 12 different types of primary immunodeficiency disorders who were treated with virus-specific CTLs. And 30 of those patients had received donor-derived, and then 6 patients had received third-party, partially matched CTLs. They had vast majority of them had been in the post-transplant setting. So half of those patients had received unrelated, match-unrelated, or mismatch-unrelated transplants, and about a quarter were haploidentical. Overall, 27 of these 36 patients had active viral infections at the time of transplant. And here is the overall response rates. So against CMV was excellent. 6 of 7 patients had partial or complete responses. EBV also was very good, with 15 of 19 having partial or complete responses, and a smaller number against adenovirus and HV6 also had good responses. The total dose of CTLs varied, with a median of 20 million per meter squared, and median time to response was roughly 28 days, with anywhere from 2 weeks to 3 months before viral response were seen. In terms of adverse events, 4 patients had graft-versus-host disease following CTL therapy. It was generally low grade, though, and treatment responsive. So 2 groups have also focused on the use of antiviral CTLs in the setting of solid organ transplantation. And this has mainly been the groups of Drs. Hake and Crawford in Edinburgh, Scotland. They previously used autologous EBV CTLs that were generated from recipients of solid organ transplantation before they underwent transplantation and immunosuppression. And then if they developed PTLD in the post-transplant setting, they could be given back these cells to potentially to target EBV. So several small studies were performed and showed that autologous CTLs were actually effective in this setting. So one group in Italy actually performed this in a recipient of a cardiac transplant. And then more recently, the same Edinburgh group had performed a phase 1/2 trial of third-party EBV-CTL that were partially matched with recipients of solid organ transplantation. And they had a reasonable success rate of 52% overall complete response or partial responses. And it should be noted that these patients had all essentially failed either reduction of immunosuppression or rituximab therapy. So these were all fairly advanced patients, and that's the 52% actually is quite good compared to what the standard response rate of clearance of PTLD at that point would have been. And also of note, in all these cases, the EBV CTL were successfully given with calcineurin inhibitors still on board, so they were able to treat right through immunosuppression. So in terms of extending donor sources, previously one limitation that's been fairly substantial has been the fact that we could only generate virus CTLs from donors who were immune to these viruses of interest. So this unfortunately excluded adults who were CMV naive or cord blood as potential as a potential source for virus-specific CTLs. However, new manufacturing protocols can now produce virus-specific CTLs from these virus-naive sources. So Patrick Hanley and colleagues at Baylor College of Medicine showed that by using cord blood-derived antigen-presenting cells, including dendritic cells and lymphoblastoid cells, that were transduced with the same PP65 pseudotyped adenoviral vector that was used in previous trials, could actually produce virus-specific T cells from the 20% fraction of cord blood. So in total, since it requires also production of the antigen-presenting cells, this whole process generally takes a minimum of 7 to 8 weeks, and thus has been best used really as a preventative measure. So this is currently an open phase 1 protocol at Baylor College of Medicine as well as at Children's National for either prophylaxis or treatment of EBV adenovirus or CMV. These cells can be given after day 28 after a cord blood transplant. Patients have to have they cannot have high-grade graft-versus-host disease or requirement for high-dose steroids. And they also have a minimum total cell number in the cord blood, and the cord blood unit has to be fractionated in order to use this 20% fraction, as shown here, in order to generate these T cells. So, in order to compare these final products from either peripheral blood-derived T cells versus cord blood-derived T cells, different lines were generated from either adults or from cord blood. And as shown here, both of these, this trial is a dose escalation trial. And as you see here, the phenotype of these cord blood-derived CTLs is generally fairly similar to those derived from peripheral blood, with a variable CD4 to 8 ratios, but detectable central memory, as well as effector T cells, and minimal outgrowth of NK cells or B cells. And using interferon gamma ELISpot, the vast majority of these cord blood-derived CTLs do show specificity. So currently, these are in a phase 1 trial, and so the majority of patients today have been treated prophylactically. There's been, I can say, there's no graft-versus-host disease seen in patients at either site, to my knowledge. And the final data on antiviral response is still, unfortunately, in the works. But I can mention that one patient at Children's National did have active adenovirus at the time of infusion, and she was able to clear adenovirus from her blood, but still had persistent stool shedding after infusion. So to date, she appears to be a partial response. So one key question is whether or not the usage of that 20% fraction may potentially compromise engraftment following cord blood transplant. This was evaluated again at Baylor College of Medicine, and it was noted that there was really no clear adverse effect of giving 80% versus 100% of a cord blood unit on kinetics and neutrophil engraftment. And I wouldn't necessarily take from this that giving 80% is any better than giving 100%. So that p-value is probably a little spurious. Additionally, we were interested in seeing whether or not cord blood-derived T cells are phenotypically different than adult-derived T cells. And then certainly T cells from cord blood are certainly more immature in many ways. And indeed, with regard to the targeted epitopes on CMV, the cord blood-derived cells do not recognize previously described dominant CMV epitopes. So for instance, here in an HLA-A02 positive cord, there was no detection of the dominant NLV marker on CMV, PP65, but instead an atypical epitope, which is shown here, starts with MLN, was recognized as one of the atypical dominant epitopes seen by these cord blood units. So this was the case for nearly all of the cord blood-derived CTL lines, that subdominant epitopes are recognized, which either may reflect the immaturity of the cord blood-derived CTLs or maybe an in vitro artifact. And we're continuing to actually explore whether or not that has any implication clinically. We've also shown through TCR beta sequencing of cord blood units versus cord blood CTLs versus peripheral blood-derived CTLs that cord blood-derived CTLs actually seem to be more more genetically diverse than those derived from adults. So here I'm showing you the TCR V-beta gene usage of a CTL line that's derived from a cord blood unit versus derived from the peripheral blood of an adult. And it's fairly obvious to see that the V-beta usage is far more diverse in the T cells derived from cord blood. And this may likely tie in to the recognized epitopes and the differences between that and adult-derived CTLs. So given the success of producing cord blood-derived CTLs, we also extended the system to see if we could derive CMV-specific CTLs from adults who are CMV naive, which depend on specific ethnicity, but are a fairly notable population. And this would potentially expand this treatment to patients who receive bone marrow transplant from CMV naive adults, which are at particularly high risk of having reactivation, particularly if they've previously been exposed, the recipient has been exposed to these viruses. So to test this, we sorted the T cells in order to isolate naive T cell populations via CD45RA from CMV naive adults, and then used otherwise the similar processing to that used for cord blood derived CTLs using donor derived antigen presenting cells as well as lymphoblastoid cell lines propulsed with CMV peptides. And this thus avoided antigenic competition from adenovirus or EBV, which are far more ubiquitous than CMV in the general population and would potentially outgrow CMV when you're attempting essentially a primary immune response ex vivo. So this actually did result in PP65 specific CTL from these virus-naive donors in a similar time frame to that of the cord blood-derived CTL, so generally 6 to 8 weeks in total processing. And they were similar phenotypically to peripheral blood-derived CTLs in their epitope specificity. And the phenotype of these cells was predominantly CD8 positive cells, which is what we would mostly expect for CMV-specific cells, with detectable central both effectors and central memory cells, as shown here. And despite the various successes in generating virus-specific CTLs from various donor sources, there are still many drawbacks to using a customized product. So obviously you require an available donor, so it would not be helpful for patients undergoing gene therapy who have viral infections, or it wouldn't be helpful for those who had undergone cord blood transplant where that 20% fraction is not available. There's also a major time commitment to generating these custom products, a minimum of 2 weeks for virus-exposed donors, not including testing time, and then 6 to 8 weeks plus for virus-naive donors. So obviously, in a patient who is critically ill, this is prohibitive. So third-party partially treated CTLs surmounts these issues by using potentially off-the-shelf therapy for persistent viral infections. In order to do this, obviously, knowledge of the MHC restrictions for specific viral epitopes is required in order to ensure that the recipient and the donor of those CTLs share at least one HLA allele that mediates antiviral activity. And this can be done in various ways, either using pentamers or using individual peptides in ELISpot assays that have been previously described to be restricted by a specific MHC type. So can third-party virus-specific CTLs restore antiviral immunity following cord or hematopoietic stem cell transplant? Indeed, this is what investigators at Baylor with collaborators at 7 other sites evaluated in the Chalice study. So it's most closely HLA-matched allogeneic virus-specific T cells to treat persistent viral reactivations. And here you see the idea behind. So in the study, 32 CTL lines were used to treat 50 patients who had had one or more persistent viral infections. And in each of these donors, they identified specific MHC restrictions for each of the individual viruses. So for instance, this donor, who ended up being what they call kind of colloquially referred to as kind of a super donor because he had a lot of very well-defined emission restrictions, was able to treat 3 different patients. So here, for instance, the first patient, they treated for EBV and were able to treat him with this line mediated through B08. The second had CMV, was treated with the CTL product, and the antiviral activity was mediated through A24. And then the third had adenovirus, which was mediated through A01. So here is the overall response rate for that study. And the overall saw efficacy in 74% of all patients treated. And by virus, it was 74% in CMV, 67% in EBV, and 78% in adenovirus. So overall, quite successful. And in spite of the partial match, there was little to no notable adverse events. So here are data from other third-party CTL studies, including the group of Dr. Richard O'Reilly at Memorial Sloan Kettering, who utilized third-party EBV-CTLs in several patients who had undergone cord blood transplant and then had EBV-PTLD. So he with several infusions of a partially matched product, had a response rate of 80% in 5 patients. And then 2 other studies also utilized third-party selected cells for treatment of viral infections post-hematopoietic stem cell transplant, utilizing selected cells using the gamma capture system. And they all overall also saw reasonable response rates with 3 of 4 complete responses against CMV and in 1 patient with EBV. So although, again, that would require an available donor, that also seems to be an effective measure. So here I'm showing one patient from from Children's National who had third-party CTLs for adenovirus shortly after transplant for IL-10 receptor deficiency. And he was Adoptive refractory and actually had respiratory symptoms. And after T-cell infusion, had a very rapid reduction in his adenoviral count, and we were able to detect T-cells post-infusion. So clearly it actually does provide a rescue therapy. And also mentioned that there actually are some rare cases where this was actually used pre-transplant for primary immunodeficiency, including 1 patient in the UK. And I'll rush through this because I'm running low on time, but this is a patient with CTPS1 deficiency who had EBV-PTLD pre-transplant and had Rituxan and COP therapy and then 2 doses of a third-party partially matched line. And EBV was cleared, and then the patient was able to tolerate preconditioning and cord blood transplant and did well. So this actually is a nice proof of concept for this time point. So in conclusion, hope I've convinced you that third-party cells are feasible, have low attributable toxicity, and potentially is a worthwhile treatment for refractory viral infections. And there are several prior studies that are available to date at various centers, including we have one for primary deficiency patients prior to transplant. There are third-party, as Dr. Leen mentioned, both at our center and at Baylor and Memorial Sloan Kettering. And there are also several EBV-associated lymphoma trials that are sponsored through St. Baldrick's and COG. And then I'll rush through this, but there are, in terms of future therapies, by genetic modification, there are potentially other methods that may be able to further improve antiviral CTL therapy, such as induction of calcineurin inhibitor resistance, TGF-beta resistance, and potentially also safety switch via the Cas9 inducible apoptosis system, which was in the New England Journal last year. So with that, this is the SETI group that's led by Dr. Ballard at Children's National, as well as our collaborators. And I'd like to thank Dr. Ballard, as well as our lab and our collaborators at NIH, at Baylor, as well as the Jeffrey Modell Foundation and our other supporting foundations. So I'm happy to take questions.